Okay, so this is take 3, so hopefully it all works properly. So yesterday, or the last lesson, we should have learnt about enzymes. As we know, enzymes are biological catalysts. They can either be catabolic enzymes or anabolic enzymes. So just recapping, catabolic enzymes, they break down Okay, so catabolic enzymes, they break down a substance. So I've got a one substance, which is rather large, okay, and it goes into the enzyme. The enzyme breaks it down into two smaller substances. So here's one, here's another. That is what a catabolic enzyme is. Anabolic goes in the opposite direction. Anabolic takes two smaller substances. It undergoes a reaction with the enzyme and produces a larger product. So it goes in the opposite direction. Meanwhile, when this is all happening, the enzyme remains unchanged. So the enzyme remains unchanged, even though the substrates are changed. Enzyme stays the same, and the active site stays the same. So today we're going to be learning about what happens, what factors affect enzymes. So the first factor we're going to cover is temperature. So temperature and enzymes. How does temperature affect enzymes? So enzymes move faster at higher temperatures. Therefore, they have more collisions per unit time. Therefore, there is a higher reaction rate. Let's just write reaction rate. Oh shoot, bugger. Sorry, technical difficulties. So therefore, there's a high reaction rate between substrates and the enzyme. Okay, so there's a high reaction rate between substrate and enzymes because both the enzymes are moving faster and the substrates are moving faster. Therefore, there are going to be more collisions. So this is all collision theory, which you should have covered last year, and in chemistry if you're doing chemistry as well, and probably even physics as well. So it's a very, very wide concept. So the other concept is, at high temperatures, above 45 degrees, for most organisms, the enzymes denature. Okay, so enzymes denatures. They lose shape and the active site. So that means denatures means it loses shape. When it loses shape, the active site can no longer function. So that's already that's already got a bracket around. So at high temperatures, the enzyme denatures. Okay. What I mean by denatures is the enzyme loses shape and the active site no longer functions. So we're just going to have a look at this very shortly. At these temperatures, enzymes do not function and can no longer function. So that's the main difference between freezing and denaturing. Okay, if I freeze something, the enzymes are moving too slow and the substrates are moving too slow that there are no collisions happening, so there's no reaction. But if I denature something, the enzyme breaks and it can no longer be fixed. It is broken. So it's better to freeze something than to probably cook it, if you know what I mean. Okay, so if we could just wait for a bit until everyone gets these notes down, that'll be cool. If everyone's finished, I'm going to move on. So yep, okay, moving on. If you want to pause this, you can pause it now. So here is a graph of enzyme activity versus temperature. So when So when it is freezing, okay, or yeah, let's just say it's freezing, okay. But remember, there are many bacteria, many different enzymes that can actually survive below freezing. But for this example, we're just going to call this freezing, okay? We'll just call this a regular enzyme, okay? A regular enzyme that occurs within like either a human body or a plant's body, okay? Or any other kind of non-extremophile. And we'll be looking at extremophiles later on. So at 0 degrees Celsius, you can copy down this as well, okay? Enzymes don't move. So say don't move fast. So no reaction / activity. Oh no, going too low. So there's no reaction and no activity at 0 degrees Celsius. So as the temperature increases, so it increases pretty much steadily. Like it's pretty, like, I wouldn't say it's linear, but it's pretty linear. Looks pretty linear at this point here, but it increases. So at this point here, temperature is increasing. When the temperature increases, the speed of enzyme and reactants, they increase as well. And when the speed increases, there are more collisions per unit time. Okay, I'm just going to write per time. Okay, more collisions per unit time. If there's more collisions per unit time, then there is increased reaction rate, or how this is written down is enzyme activity. That increases as well. Okay, so the arrows, they just mean increasing. Okay, arrows up just means increasing. Now this point here at the top, we call this the optimum temperature. Optimum temperature, okay, or the optimum, just optimum conditions, okay. So at this point here, the enzyme is moving fast. There is the highest rate of reaction, okay. But unfortunately, if we go slightly above the optimum temperature, so around 45 degrees, if we're looking at a standard human or plant enzyme, what's going to happen is we are going to get denatures. We actually call it denaturation of proteins, but that's, yeah, let's call it denaturation. I always find it's a very funny word to say. Denaturation of proteins. But denaturation of enzymes. But most enzymes are proteins, so saying denaturation of proteins or denaturation of enzymes, same thing. Here we go. So yes, please get this down as well. I'm sorry that the handwriting's quite messy, but hopefully we can all make this out. And hopefully the video is okay. Or else you might just have to listen to the sound of my voice. And going to move on. So pause if you want to. Copy down. Okay, so here is our active functional protein. So this is our active functional, we can call this an enzyme as well. Well, if it is an enzyme, remember proteins make up pretty much everything within the cell. They make up the organelles, they make up all those little moving parts, the tubules, the microtubules. So proteins are pretty much everything well, quite a lot of what the cell is other than the cytoplasm. Everything else is kind of like a protein or something else. Well, anyway, so these enzymes, they're active. Remember, at the start, they are all wrapped around, and there is a specific site. Okay, there's a specific active site in which the substrate can bind. Once there is a high temperature, okay, so, oops, keep on moving, okay, so there is a denaturation at high temperature, okay, a denaturation of high temperature. It pretty much breaks, okay, breaks bonds, okay, it breaks the bonds holding the protein in the specific shape. Okay. Remember this word protein. I might use it sometimes to describe an enzyme, because all enzymes are proteins, but not all proteins are enzymes, if that makes sense. Okay. So this is what happens. Remember, this protein has a specific shape, and that shape gives it its function. But if I completely lose the shape and it becomes denatured, it has no function. It can no longer work. The other thing about denaturation, a denatured protein can never go back together. Okay, once it's broken, it's broken. It's like when you fry an egg. If you fry an egg, you're denaturing all the proteins within the egg. But you can't take a fried egg and put it back into a raw egg. It's not possible. However, if you freeze an egg and then you thaw it out, it theoretically should look just like the normal egg that you put in. But I've actually never tried doing this, so don't take my word on it. Okay, moving on. So, don't need to copy this down, but here are I'm just going to give you a couple examples of these things called extremophiles. Extremophiles, so that's an I. Extremophiles. So those people who know Latin, what do you think extremophiles mean? So we already know philes means love, okay, or extremely like. So we're looking at those hydrophobic, hydrophilic. So phile means you like them. Extreme is extremo, extreme. Okay? So extreme lovers. No, not what you're thinking. But yes, they're extreme lovers. So they love the extreme conditions. So a Methanopyrus candeleri, okay, that's a bacteria. It can survive and reproduce at 122 degrees Celsius. So where do you think these environments which are 122 degrees Celsius on Earth? You are correct. So it is 122 degrees Celsius underwater by hot water vents. Hot water vents. That's where you'll find this bacteria, and this is where this bacteria actually produces. It's a really cool. It's a really cool bacteria, I guess. I mean, it's living underwater, so that's where the underwater, like near the bottom of the sea. So the pressure is massive. It's getting hardly any sunlight. It's actually eating the methane. Okay, so the methanopyrus. Okay, it actually consumes the methane around it to survive, and it survives in such high temperatures. So that's one of those really cool bacteria, and it's an example of an extremophile. These are not tested, so don't worry too much about it. But it just shows you that there are enzymes within this bacteria which can survive denaturation. So it is possible for some enzymes to survive past that 45 degrees Celsius and be specialized to keep the organism alive in extremely high temperatures. Now, there's other extremophiles which go through very, very low temperatures. So the Grilloblattidae, okay, or ice crawlers, have an optimal temperature of 1 to 4 degrees Celsius. Now that is very, very cold for an insect. So again, these insects, okay, they have special enzymes within their body which don't freeze up, okay, which can still work at very low temperatures. So that's another extremophile. Yeah. Okay, and this is just a picture of my friend. She actually went to Antarctica. She's doing a I think she's completed her PhD, but I'm not too sure yet. But yeah, she was doing I think she's nearly finished her PhD, and her PhD was to actually go to Antarctica, look for bacteria. Okay, so look for bacteria in the Antarctic snow, and find those bacteria, extract the enzymes, and test their enzymes, and test like what temperatures do their enzymes work, and actually molecularly break down their enzymes and break down the bacteria, and have a look at their DNA and like do some sequencing. So that's one of the really cool things about New Zealand, is that we are kind of also in charge of Antarctica as well. So you can have a lot of interesting research opportunities if you pursue something in biology in New Zealand. So yeah.